now we're going to move to the first panel on brain cell atlas data annotation and integration, where the panel will address 4 topics: how to generate an annotated and implemented atlas from current atlas data for use by the community, how to link these atlases to other species, over development, and integration of spatial data. So this panel is moderated by Drs. Aparna Bhaduri, who's Assistant Professor of Biological Chemistry at UCLA, and Rahul Satija, Professor of Biology at NYU.
Okay, well, thanks very much, Amanda, for the introduction, and for the NIH for inviting Aparna and I to moderate the session, and to everyone who's volunteered to serve as a panelist in the discussion. So, Aparna and I will just give a very brief introduction. I mean, we obviously had a beautiful introduction from Ed and Hongqi and Bing and Joe about the value of being able to create a brain cell atlas. And of course, almost basically all of us in this community are very familiar with both the incredible challenges required to build this, but of course, the tremendous opportunity if we're successful. And just to briefly summarize, I think having a single reference framework to be able to broadly interpret different types of data in the mammalian brain would be transformative, especially for integrative and comparative analysis. Speaking very simply, having a common reference framework allows us to learn from each other's data. So some of the questions you've already heard about today, or we'll be continuing to hear about, for example, trying to understand how the spatial organization of cell types varies across species, in development, how multipotent progenitors progressively specify and choose their lineage, across aging and neurodegeneration, which cells are vulnerable and how they change their states. But basically, all of these types of questions and many more require some degree of common nomenclature or a common language or a common reference framework for us to be able to talk about and interpret data. And that challenge of being able to build a single uniform reference is something my lab has been very involved in the Human Biomolecular Atlas Project, or HuBMAP, where we've developed a single cell reference of the human body, which we call Azimuth. And that's been really transformative for us for being able to integrate data across HuBMAP sites, but it's also been really rewarding to see in the community to watch other people adopt that reference so that they can compare and contrast both with their own data and with HuBMAP data. And of course, Bakken has made extraordinary progress in this area, and in fact really shapes our view of how we view this in HuBMAP. But the biggest challenge is the same as the opportunity, is that we need to now be able to collapse many different data types together into a single uniform framework. And I think that raises both technical challenges, how can we take spatial and single cell, and how can we take, different modalities and name them and integrate them in a robust way? So there's technical challenges, but there are also sort of broader conceptual and community challenges, how we can agree on a particular set of names, and even like whether we should be naming things the same across different species. Of course, the cells are not identical, so it raises a lot of conceptual questions for how to do this. And so we've tried to structure the discussion today and some of the questions to capture both these sort of like core technical challenges, but also the conceptual ones as well. So I'll turn it over to Aparna, who will tell you a little bit about how we're structuring the session today.
Thank you, Rahul, for that great introduction. And, I know we have a few technical issues, so I'll try and go through this quickly. But basically, the goal for today is thinking about how to collaboratively construct a reference atlas, leveraging these atlases for cross-species analyses, linking atlases across developmental time points, and thinking about how to collaboratively construct a reference atlas, possibly using tools like spatial information as well. Each of these topics is going to coordinate with a speaker and then an open discussion. And for the first topic, some of the key discussion points will be about constructing this core reference and thinking about community agreement and governance. For the second, when we're thinking about other species, we'll really be thinking about cross-species annotation challenges, as well as whether there are key community standards we should be thinking about for multi-species taxonomies. For development, we're really thinking about core challenges for annotating developmental data, as well as community adoption. And you'll notice that there's some patterns here. And then in terms of the spatial analyses, core challenges for annotating spatial data and community harmonization. So with that, we look forward to a great open discussion, as well as key points that will be brought up with each of our speakers. So I'll turn it over to Rahul to introduce our first speaker.
All right. Thanks, Aparna. So very grateful to 4 speakers who have agreed to give short talks on their work, which we hope will help kind of illustrate some of these questions and see discussion. And then after they speak, we'll just have an open discussion seated by some questions. And our first speaker is Scott Daniel, who is a software engineer at the Allen Institute for Brain Science and their information architecture team. And Scott has worked on the Map My Cells tool, which you heard about earlier, is a really extraordinary software package that just takes single cell data from a variety of sources and just magically maps it into a reference atlas. My lab uses this all the time, so do many of yours. So thanks very much, Scott, for helping to build this and for agreeing to give a talk here.
Yeah, thanks for the opportunity. As Professor Satija just said, my name is Scott Daniel. I'm a data scientist here at the Allen Institute. And so yeah, I'm talking about Map My Cells, which was our attempt at the Allen Institute to start to build an answer to this question: how do we get everyone talking about the same thing in the space of reference atlases and reference taxonomies? Our goal was, our thinking was, if we could get everyone to start annotating their data to shared taxonomies, then we can start putting data sets and studies in conversation with each other, because in theory, we'll know when the data is referring to the same biological truth, whatever I mean by that. There are challenges involved. There are many ways to solve this problem. And some of them are very complicated. They involve complicated infrastructure. They involve complicated hardware. They involve models that need to be trained over days. And so what we at the Allen Institute wanted to do is just provide a tool that was lightweight, that anyone anywhere could use. They just put in their data, and without thinking about it, they get an annotation back, and then they can move forward with their analysis. And so what we ended up producing was a web app, which hopefully many of you are familiar with, which does just that. You upload your data, 5 to 20 minutes later, it is annotated according to one of the taxonomies supported by Bakken, and an open source Python library that provides the software running at the back end of that app. And this is for people who want to download and run it on their own machine, if that is what they want to do. The web app supports 4 publicly available taxonomies: the whole mouse brain taxonomy from Yao et al. 2023, the whole human brain taxonomy from Saletti et al. 2023, the cross-species HMBA basal ganglia taxonomy that will be published later this year, and then a human medial temporal gyrus taxonomy from the Seattle Alzheimer's Disease Atlas Consortium. And these are the taxonomies that are supported by the web app. If you download the Python library, install that, you can, in theory, pre-process and map to any taxonomy that you have access to. And the idea, and this is kind of feed into our thinking of Map My Cells as a framework, a place that people can bring their taxonomies and their algorithms and allow each other to use those to annotate disparate data sets. To help motivate the discussion, I'm just going to give a very cartoonish description of the main algorithm running in Map My Cells. This is the hierarchical clustering algorithm. What Map My Cells wants to do is take your data and for each cell annotate it at each level in a hierarchical taxonomy. So it does this using a pre-specified set of marker genes. For every level in the taxonomy, it takes those marker genes, randomly subsets them, uses that random subset of marker genes to correlate the cells against the cell types, chooses the cell type that is most correlated with your cell, and then repeats that process 100 times, each time using a different random subset of marker genes. It keeps track of how often each cell type is chosen and then assigns the cell to the cell type that was chosen the most frequently. What this allows Map My Cells to return to you is an annotation for each cell, but also a confidence score. You can now say, like, if the same cell type was chosen 93 out of those 100 iterations, you can be pretty confident in that mapping. If it was only chosen 51 out of 100 iterations, maybe you treat that cell with a little more caution. You also get metadata telling you what version of Map My Cells was run, what configuration parameters it was run with, and what global set of marker genes Map My Cells used, if that is important to your science, which it usually is. So that's what Map My Cells does. Is it worth doing that? To evaluate Map My Cells performance, one of the first things we did was a standard test train holdout evaluation. We took the single cell RNA sequencing data used to define the taxonomies and split them into a training set used to derive marker genes and average gene expression profiles for cell types, and a test set. And then we mapped the test set back to the taxonomy and compared the labels derived from MapMyCells with the de novo labels in the test set that the original authors gave it. And we see a very good correspondence. These are the F1 scores of those mapping. And it really only starts to degrade at the lowest level of any given taxonomy, which in many cases, some people argue is more of a computational construct and has less biological meaning than the higher levels of the taxonomy, where the expert annotators have grouped the computational clusters into meaningful neighborhoods and groups and such. We also ran this test on the Langley et al. 2023 whole mouse brain taxonomy published by the Broad Institute, which is not an on-label taxonomy. It's not one of the taxonomies that MapMyCells was designed for. So this is a case where we took the open source Python library, pre-processed another taxonomy, did the test train holdout, and still got very good mapping. Again, which is kind of an argument for MapMyCells as a framework or a mindset rather than just a set of taxonomies you can map your cells to. So that addresses the accuracy of MapMyCells. Is it trustworthy, right? Which, I mean, does MapMyCells do a good job of reporting when you can trust a mapping and when you cannot? And I would argue that it does. These plots show cumulative distributions of that confidence mapping, that fraction of those 100 iterations that pick the same cell type. The orange distribution is for cells that were correctly labeled, and the cyan distributions are for cells that were incorrectly labeled. Top row is whole human data, bottom row is whole mouse data. And the big takeaway is that these distributions are very different. At each level in the two taxonomies, there's a cut you can make that discards 60 to 70% of your incorrectly labeled cells while only sacrificing 10 to 15% of your correctly labeled cells. So that quality metric really does allow you to, look at the mapping and say something about, do I believe this or do I not? So up to this point, we're evaluating MapMyCells on its own terms. We've been using the RNA sequencing data that was used to derive the taxonomies MapMyCells maps to. But what's going to happen in the real world when people bring their own data that might have different signal-to-noise properties than the data MapMyCells was trained on? To evaluate this case, we went into the real world and found other data that was out there. We went to Cell by Gene and got a mouse attack-seq data set from Lee et al. 2021 and mapped it to our whole mouse brain taxonomy using MapMyCells. The confusion matrix on the left shows the correspondence between the cell type classes found by MapMyCells on the horizontal axis, and the vertical axis is they called it subclasses in the original publication. And you can see there is a strong diagonal component, which gets stronger when you make one of those quality cuts I alluded to earlier. So the confusion matrix in the middle, that is only accepting cells where 95 out of those 100 iterations chose the same cell type. And you can see that most of the noise from that confusion matrix is gone, and we only sacrificed about a third of the cells. The final validity check we ran, the histograms on the right, are taking the data and subdividing it by anatomical dissection and comparing in orange the distribution of subclasses found by MapMyCells with in blue the distribution of the subclasses in the original training data for those same anatomical dissections. The idea being, if MapMyCells is finding biological truth, these distributions should look the same. And qualitatively, they do. You see spikes in the same region of subclass space between the two distributions, indicating that MapMyCells is finding the same distribution of types per anatomy as was in its training data. We also grabbed spatial transcriptomics data from the wild, from this Xi et al. 2023 publication, and again, mapped it to the whole mouse brain taxonomy. All I really wanted to say about this plot is these are the cells colored by the classes and subclasses that MapMyCells found in representative slices of the spatial transcriptomics data, and you can see that the cell types trace out the biological structures we would expect them to trace out. So again, indication that MapMyCells is finding the biological truth. And the final feature we wanted to test was we now have the ability, using just gene ortholog tables published by NCBI, to let users map across species, to bring, for example, rat data and map it onto the whole mouse brain taxonomy. Or in this test we did, we took the whole human test data and mapped it onto the whole mouse taxonomy. And this confusion matrix shows the highest level of the two taxonomies, the vertical axis being the ground truth human annotation, horizontal axis being the mapped mouse annotation. And again, strong diagonal component that makes sense. You get excitatory neurons mapping to excitatory classes and non-neurons mapping to non-neuronal classes in the whole mouse brain taxonomy, which just kind of shows like this is where we want to be going. This is where people bringing their data in and annotating them to these agreed upon taxonomies in a way that we can put the data in conversation with itself. thank you for your time. Thank you for this opportunity.
Thank you, Scott. That was lovely. So maybe I'll ask an initial question, and then maybe Scott, if you want to take a shot at it, and then if other people would like to jump in, that'd be wonderful. If that sounds good. So I like one of the things you said at the beginning, which was that you want this tool to be something that users don't have to think about, which is sometimes a little scary to say computationally, but that goal of just sort of one click, there's a pretty strong precedence for this. I like to draw an analogy to genome read mapping. We don't, we just map our reads. We don't have to make that a major intellectual exercise every time we look at short read data. And we want single cell analysis to eventually reach that level of robustness. And so it's interesting to think about the similarities and differences between those two problems. And I would argue that one thing that the genome people have that we haven't quite gotten to yet, well, a couple things are one, you talk a lot about uncertainty and confidence, which is really valuable for users to look at, but we don't necessarily have something like error, like an eval that says how different is this cell compared to what we think the reference should be like. And that's a really difficult metric to derive, but I'd like to open that up if you or others have ideas for kind of if that's something we need and how we could do that. And then the other is that, of course, the genome communities have these really strong, sort of well-established pipelines for updating sequential references over time. And that's what my lab has found really difficult. It's not just building the reference the first time, it's sort of continually updating it and adapting it to new data, which can sort of take years or even decades of time. So those aren't easy things, but I wanted to hear your thoughts on that. And then maybe if other people want to jump in as well, that'd be great.
Yeah, so the error, quantifying error is definitely an open problem that we are still working on. In addition to the quality metric I mentioned, there's also just a Pearson's correlation coefficient, between the cell and the chosen cell type in the space of marker genes. So we have those two metrics, and one of them, and there's an interesting interplay between when you use one and when you use the other. But yeah, getting a quantitative, like what does it mean when only 60% of the iterations chose the cell type? Does that mean the cell has a 60% chance of being correct and a 40% chance of being wrong, or is it non-linear? It is an open research question that we're working on. That's that's kind of a non-answer. I'm sorry for that.
It's really hard also because a cell could be exactly what you think it is, but only because there are 4 or 5 marker genes that we really know and we ascribe biologically to be canonical, but that might result in a very poor overall Pearson correlation coefficient. So I don't have easy answers to that one either. I think people are raising their hands, so that's very helpful. So maybe Mike from the Allen, if you want to jump in.
Yeah, thanks, Rahul. I mean, I guess as Scott says, there are many methods for doing this. If you look in the literature now, there's there's many types of algorithms of varying degrees of sophistication, and I think that you can't. What we can't really rely on ultimately is that you're not going to answer these questions by marker genes and gene expression alone. We have to bring in other modalities, other information in identifying these types and mapping. There is no real ground truth when it comes to using only the transcriptomics alone. So I think that more as we, increase the ability to leverage these other modalities, I think we'll start to get stronger identification.
Can you say a bit more before we move on about what other types of data, maybe it's functional, maybe it's electrophysiology, are you most excited about that you feel will be helpful? Because I think we all agree that RNA is a limited subset, although a valuable one. And it might be a modality that you guys are good at measuring, or it might be a modality that you really hope that someone in this community will figure out how to measure at scale.
Yeah, I think it could be things, and perhaps Trygve has ideas there. I think I'm channeling him, I guess maybe, but I think the epigenetic data, the spatial data, other data types, things that can lend. We ground it in the transcriptomics, but on the other hand, there's other supporting modalities which will serve to give distinction, and even other aspects of literature too. So it's early days, I think, in accurate cell type mapping.
Trygve, you want to go ahead and jump in?
Sure. Yeah. No. I agree. It's early days in having a community reference, and I guess one hope is that we can have bound cells be more of a two-way street, where as people bring new data sets, maybe they're looking at a disease state where it's a similar brain region, but things are slightly shifted, that people can have capacity to sort of annotate the reference itself with the various metadata of the cells and donors that they're bringing to bear. And I think this will certainly be useful in bringing those additional data modalities to bear. And so, even if it's, oh, this mapped very well and the spatial distribution makes sense, I have this extra information. And that can be sort of collated in addition to sort of the sort of agentic AI-based lit searches that may, generate a bunch of potential annotations. And then all of that can maybe be rolled up into version releases of new taxonomies to facilitate better mapping in the future. And I think that's also going to be important for as people bring their sort of less common but very important model species. And we'll probably get more into this later, but I think that's sort of the other side of the coin of these mapping approaches.
Zijan, would you like to unmute and jump in?
Yeah, I want to touch on the problem of how we reconcile different versions of the references. I think maybe this is the goal of doing all this exercise across species, across modality, and across development time is to try to get to the consensus, and we try to reiterate all the versions of taxonomy, and especially for the higher level of taxonomy to be as robust and reproducible across all this as possible. And then leave all the discrepancy and how to describe things at the lower levels of the hierarchy. So that might be our strategy, is try to reconcile this, is to take whatever is most robust on the top level of the hierarchy so that people see them over and over and over again. Yeah.
I think that's one thing that's really nice about what the Allen has always done in their hierarchies is make them hierarchical, is that it's not just a single level, but you have this ability to drill down, and you can sort of assign your level of confidence based on how deep you think the data is willing to go, and that helps sort of build confidence over time. I think that question of how you decide on robustness and how you reconcile across hierarchies is a really nice thing to push further on. I might call on a couple of people just because I'm curious. I see Bing is a panelist, and I also see Jesse Gillis, who I know works on this as a panelist. So while waiting for either of you to jump in or unmute, I'll just say that one of the things that I've tried to sort of argue for a little bit in HubMap is, even if we can't get things perfect, as long as we're calling things the same thing, that's better than having nothing and sort of, everyone having their own sort of individual taxonomy that they agree in. As long as if we're working together towards reconciliation, but even in an intermediate stage can have a set of broader labels that we can call data the same way, that's very, very valuable.
And while we wait for those folks, I see that Vatsala has had their hand up for a bit.
So I had a question where you said you used this program to map the different regions of the brain. So you said that showed real life proof that it's correctly genotyping the cell type. But I have a question, like if a neuron, if it's a state, like let's say it's an astrocyte or a glial cell, if it's expressed in a certain brain location, is it going to have different RNA-seq data? So if you have the same type of neuron, but expressed locationally in a different area, how are you mapping that?
I can comment on that. I think we do have cell types that share across the whole brain. And if they don't have any signature, if you don't have any differences, then it's the same cell type no matter where you are. But in most cases, like astrocytes, they do separate particularly at a cluster level, or even sometimes at a subclass level. So you can separate them, and then they will be mapped correspondingly. They say that this is astrocyte localized in striatum, for example. Yeah.
All right, thank you.
But just really quickly on that, I think it does point to an important, maybe new feature of MapMyCells that could bring in that anatomical context to the query.
Okay, yeah, I see Jesse's raised his hand. Go ahead, please.
I'll just chime in since it's a problem, as you mentioned, we're super interested in general. Yeah, I think the point made that, we really just want to have the same name for the same thing is kind of a key point. And as long as these methods help us do that, they have some utility. And I guess I would say that I feel that, in some way, just to harken back 10 years ago, like all these methods worked, and it was surprising that they worked at all, and it was all easy, right? Like t-SNEs weren't designed to find cell types, and then they found cell types, and it was really straightforward. And then you push the methods far enough, and you get cell types that we're not sure what the word cell type means anymore. And so I feel that we've reached this stage where we're a little bit of danger of overfitting to existing data that can serve as a reference under some paradigms, but possibly not all. And then you get hints of that sometimes in the questions that were asked about, well, what does it mean? Do I have the same cell type if I have a different state? And what are we talking about when we say cell type exactly? And at what level of resolution do we expect mapping? And what do we want to be robust to and not want to be robust to? If this is all trained up on, a particular inbred mouse line, like, do we want things to be robust or overfitting to that class of variability? And so I just would say that I feel that absolutely terminology is important. Absolutely having good pipelines that map to existing references is important. And having measures of intrinsic variability like this nice approach for just bootstrapping through the marker genes is sensible. But that also, everyone in this room can, I'm sure, collectively think quite a bit about sources of variability that are not being sampled and that we risk overfitting away when we pick new references, prematurely potentially.
Yeah, I think that comes up a lot in the developmental and cross-species context that we'll talk about too. Let's let Davide and Xiaoyin chime in, and then we'll move to our next speaker.
Thanks for the very nice talk. I was just wondering if you have, as you're developing this hierarchy, if you're thinking also of integrating with the cell ontology, or if you're thinking about it in ontology term, because sometimes I feel like hierarchy is nice to think about, but then there may be more complex sort of relationships among cell types. For example, a cell may be different things. It may be spatially located somewhere as well as of a given type, of a given state. So I was wondering if that's something on your radar.
I'd just like to comment that we did do that in the Bakken basal ganglia data, where David Osumi-Sutherland and others worked toward understanding, the relationship of these cell types with the cell ontology and extending that. So that definitely is work that's ongoing.
Go ahead, Xiaoyin.
Right. I also have a question along the line of the hierarchical structure. Zijian, maybe you can shed some light on this. I also like the hierarchical structuring and so on, so that people can get more confidence on the higher level, and then depending on what you like at the lower level and so on. But I remember in our discussions, about the mouse brain, I don't know whether that still remains true, that some brain regions seem to have more nice hierarchical structure, and some brain regions, although the diversity could be just as large, but they're more flat. And then that thus making the hierarchical structure harder to use as a way to deal with things. Is that still remaining true as you dig deeper and deeper into the data?
I think that remains true. I think we have some general guiding principle to where things belong, like whether it's supposed to be a class or subclass or lower level. Basically, I think our class levels is intended to be major brain regions coupled with neurotransmitter. That's kind of roughly how we like our class to be. And subclass is mostly driven by how discrete they look like in the map, in a way, how reproducible, like when we do cross validation, what level of confidence we can get, and transcription factors. There has to have really strong transcription factor markers that disambiguate them, and every close related types, they should be spatially segregated. So there are some, not completely automated by some like a rough, like general guiding principle of where we put things. But all this is not super principle yet. I mean, I would like to have more better ways. And also is like we also hesitate to put a whole subcortical areas, all the types that are closely related to each other into one big blob, because you also make that class to be too heterogeneous, way too many things at the same time. We'll try to partition a little bit further so that is more like a hierarchy instead of one big branch that covers everything. So yeah, so that's guided by a lot of the practical concerns. It's not purely driven by computational, how similar the cell types are related to each other. Yeah.
Okay, I think we have to move on. We're already a few minutes behind, but I think many of these questions we can talk about in the chat, and of course will come in and they're relevant to our next topics as well. So our next
Rahul,
speaker...
I'd like to acknowledge your early leadership in the development of these mapping approaches and the work that you did with Azimuth and stuff, which has played a very important role. So
thank you, and which we did in close collaboration, of course, with you guys. So our next speaker is Fenna Krienen, who is at the Princeton Neuroscience Institute. And I first encountered Fenna when she was a postdoc, I think, in Steve McCarroll's lab and published, I think, some really foundational work comparing cell type evolution across humans and primates in particular, which is work that she continues in her own lab and will tell us about today.
Thanks, Rahul. Thanks, Aparna, for organizing this. And yeah, I think you'll see in my slides, like all of the statements are more like questions. So the purpose is to really seed more discussion for the forthcoming panel. So, okay, let's begin with some more questions. Depending on how you think about cross-species cell type taxonomies, you can come at these from different angles. And as Rahul alluded to, in my lab, we're really interested in the question itself of how cell types change over evolution, but of course it's equally valid to be interested, in building a comprehensive cell type map of the human brain and to understand how those are unique, or even just the practical consideration of the challenges of measuring every cell type in the human brain and allowing species to potentially leverage, smaller brains, more deeply sampled species to help you fill out those gaps that we currently have in the really large brains. So I was thinking about these questions, your perspective and your interest in developing cross-species cell type taxonomies when putting together these couple of slides, but also some of the challenges. So really, what assumptions are we making when we integrate across species and when we infer cross-species cell types? Because it's sort of a two-step process. First, you decide how you're going to bring those two species together, and then you have to make some decisions of what you're really going to call a homology, what you're comfortable with there. And then I also want to touch a little bit on really the practical, we're midway through BICAN, we've had BICCN, we have a lot of data now available accumulating from different species and mostly the primates and the mouse. But how do we really start to leverage those in real time to fill in the current gaps in human brain profiling, which is where a lot of people are interested in going. What I won't touch on much is these broad phylogenetic surveys and profiling, which we have been doing, but I hope on the panel, maybe Morgan or Trygve or Jesse Gillis or Alex Pollen is here, will step in and kind of give some insights onto how valuable it is to have broad species phylogenies being sampled in the same way. And so what I want to emphasize a little bit about is now we're really starting to collect, as Ed and Honghui said, atlases that are approaching comprehensive profiling and approaching millions of cells. And one of the challenges of cross species is not just evolutionary divergence, but also, again, the practical challenge of sampling across space. And so we have these issues of resolution. How small can you go? The mouse brain is very small. Some of these areas are very small. We have a minimum tissue volume input for dissociated cell preps. For spatial, of course, you get more resolution and potentially more denser sampling with small brains. And in humans, it's still fragmented and incomplete, given the performant technologies of today don't yet easily scale to human brains or larger brains, but of course that's changing. And so these are the practical challenges with the data that we have in hand. And then, of course, spatial alignment and cell type alignment when we have inconsistent sampling schemes across species. One of the challenges that I hope we can pick up a little bit in the panel discussion is this pervasive issue of many-to-many cluster relationships, which I would argue, regardless of how you're integrating or aligning using map-my-cells, or other approaches, which we use all the time and is a wonderful tool, we sometimes have to contemplate not just one-to-one alignments, but many-to-many cluster relationships. How many of those are biologically real? Diversification in one species versus another? How much of it is technical artifact driven by the sampling and the resolution? And really, how do we accurately capture and distinguish real biological variation from those technical sources? And so I just love this plot from Rebecca's paper now quite many years ago. 2 different cortical areas, 2 different species. And yet you can see in some cases you have beautiful one-to-one alignment of their resolved cell types. Again, these are cell taxonomies that were independently generated per species and then mapped together. And some of them map very well. Some of them have many-to-many relationships. And again, is this because these cell types are not changing in evolution? Probably not. Is it because they're more distinctive within relative to the other cells in that sample? So distinguishing those features becomes very important. And I loved so much this visualization that Ed and Honghui showed at the beginning in their keynote. I think Trygve might have put it together originally, so I screenshotted it so that we could use it as maybe a discussion framing to think about what are the assumptions that go into doing something like this, and what is the value and what is gained. We talked a little bit about the power of having a harmonized nomenclature. So Ed mentioned that if we have integrated species first data sets together, whether they're spatial or single cell or what have you, and then we build into the naming, the nomenclature of that cell type, the criteria that whatever we name it with has to apply equally to all species, that's very powerful. Again, thinking about, though, what might be lost. Of course, if a marker is not conserved across species, we don't name that cell type that way. But that could be a very good marker for one or more of those species contained within that consensus cluster. So thinking about how we continue to represent that information. And of course, this is part of this strategy that you start with a kind of cross-species integrated data set and then allow single species clustering to proceed. So you have an initial anchor of cross-species alignment or harmonization, and then you allow the species-specific variation to proceed further down the line. Of course, that was put to the test, that principle or this approach was put to the test in the recent preprint by Nelson and others, where really taking the basal ganglia data that we've been collecting within the human and mammalian brain cell atlas from each of the primates, putting it together and doing this kind of species first approach gives us that. But of course, we have all of these amazing existing taxonomies between species, and we have these mapping tools, map-my-cells and others, and those have already been analyzed. They have beautiful hierarchies of cell types, but the measurements might be different, the species might be different, the taxonomies are different. And so, of course, map-my-cells still and other mapping approaches still becomes very useful and lets us transfer labels, as Ed alluded to, but also learn something about the older atlases or the disparate atlases. So just one example from our lab, using, again, map-my-cells to transfer labels from that initial whole human brain atlas onto our marmoset data. Using first the RNA, we can see that at these highest levels, these coarse levels in the human data, we have lots of groups and clusters in the marmoset that map onto those. If we then just focus on one of the most nebulous superclusters in that original human paper, and because we have the spatial data, we can ask now, is there actual spatial structure to all of these really difficult to resolve fine clusters within this homogeneous supertype? And indeed, there's beautiful structure, which wasn't measured in the original human data, but then the beauty, of course, is to transfer that back to the human atlas and say, okay, now we have some spatial registration. Of course, it's ported through another species, but if we believe in these harmonization approaches, we can use that information again and transfer it back and enrich the original human atlas. So again, I should wrap up soon, but I want to give one more example of, the power of maybe using these cross-species mapping to learn new biology. And I just couldn't resist, because some of these questions of cell types that distribute to other spatial locations maybe also tease up future discussion as well. So one thing we know is that in the thalamus, in the rodents, for instance, there's a real paucity of GABAergic neurons. That's not true in the primates. So in the thalamus, you have abundant GABAergic neurons. That doesn't mean they're totally absent from mouse, but there just aren't so many. And so the question is, where do primate expanded thalamic GABAergic neurons come from? There have been several existing theories, but using these mapping tools, again, not just single cell to single cell, but single cell to spatial and spatial to spatial, we are suggesting, our data are suggesting that this thalamic expanded population in primates might have originated from a really closely related cell type that is conserved in the brainstem of both species. And to this question of whether the spatial location of cell types that might end up in different places across species matters, what we see also in the chromatin accessibility profiles is that these neurons, these GABAergic neurons in the primate, retain their midbrain epigenetic identities. So this is my last slide, but again, just to seed some more questions, when we have changes across species, it might come at the population level, changing the abundance. It could change we know it changes the molecular properties of conserved cell types. The cell types move to different regions. And of course, we are looking always for gains and losses. But again, we have these challenges we have to overcome. So rare cell types might be missed if the data are not large enough. The question we still haven't addressed is, how divergent can a cell type be and still be considered homologous? And how and when should we prioritize classes of genes to assign those homologies? How do we deal with these many-to-many relationships? If we look towards whole atlas integrations, then we can find these really interesting cases of spatial relocations. And of course, this question that always comes up, what is a new cell type and how do we define it? So I will just stop there, and maybe the panel can commence.
Thanks so much, Fenna. That was a really thought-provoking discussion. One question that I have for you, because I think you did a really nice job of highlighting some of the challenges related to sampling resolution and then spatial mapping and thinking about one-to-many cluster relationships. So my question for you would be, what do you think are needed computational, experimental, or conceptual innovations that can fill these gaps to establish real biological variation? Or from the tools that we do have today, what evidence should we be prioritizing to make these decisions?
Yeah, no, I mean, of course, I don't have an answer to any of those particular dimensions, but, one thing that we should realize is that we're kind of midstream in a much larger process. So eventually we will get to whole, say, human brain sampling in the spatial domain, in the single cell omics domain. We will never get the physiology. And so the question is, how much inference can you make from observations that you make in other species? And I think what we could really use some more help with is not just saying we can measure these 2 or 3 different modalities in the same cell type or in the same species, but really make the causal links between those modalities stronger, so that that can really afford predictions in the species for which we may never be able to measure those processes directly. So that's one thing. And then the other thing that might be interesting to think about is, we're in an era of using a lot of generative models for things. So can we literally fill in gaps in unobserved data, especially say in the spatial domain, in the case of these larger brains, that can help us get closer to a unified structure to work with across species.
Super helpful and lots to think about there. I'm excited for the panel discussion. We are a little bit behind time, and given that some of the challenges are actually parallel between comparing species and thinking about development, and it's all related but different, I was thinking, in the interest of time, let me introduce Tom Nowakowski, and then we'll have a panel discussion afterwards. Tom is a associate professor at the University of California, San Francisco, and he's been very involved with the BRAIN Initiative from its initial stages and has really pioneered the discovery of transcriptional signatures that describe a lot about both human brain development as well as moving into the adult as well. He has made seminal contributions to the field in understanding novel lineage relationships for radial glia and cell types in the cortex, as well as has been characterizing cell types in other brain structures, and has been the chair of the developmental working group and really spearheaded the process for the recent brain package. So it's really an honor that Tom has been able to join us today, and I look forward to his thoughts in thinking about the developmental piece of this axis as well.
Yeah, so as we just learned, and as we're learning, the adult brain can contain somewhere on the order of 5,000 different transcriptomic cell types. And if you don't subscribe to the idea that each of those transcriptomic clusters represents a different cell types, that's still a ton of transcriptomic variation and clustering that we need to account for. And I think to us, or to me, the interesting question is where do these cells emerge from during development? What we've known for about a decade or two at this point is that most of the neural cells, vast majority of cells in the brain originate from neural stem cells, which in the cerebral cortex in particular are referred to as radioglial cells. These cells produce all cell types, the neurons, oligodendrocytes, as well as astrocytes. However, as we are just learning, thanks to the amazing work of Bakken, the ICCN, is that they are tasked with not just producing one type of each of the kinds, they have to produce many different types. Even if we look at just a small part of the cerebral cortex, such as the visual cortex, we now know that there is approximately 100 different cell types. So how do these radioglia accomplish this amazing challenge in a short period of development relative to the lifespan of most organisms? Well, what we've also learned through a variety of different experimental approaches is that one strategy that these cells utilize is that they produce different cell types at different times. And so an individual radioglial cell might go through different phases of state of differentiation competence to produce first different types of neurons and then producing oligodendrocytes and astrocytes and so on and so forth. And so when you think about this transcript in terms of transcriptomic space, you really have to distinguish two axes of variation. One, which undergoes the changes that undergo within these radioglial cells themselves. And this is a process that we often refer to as maturation, but then also transcriptomic divergence and changes that undergo or correlate with differentiation of these cells. And this very often initially involves more dramatic or radical changes in gene expression. And we essentially are dealing with very, very two different kinds of transcriptomic variation, one that occurs perhaps on a shorter time scale, one that occurs on a longer time scale. And we have to essentially factor that into our computational models. So one way in which the field has attempted to resolve this problem is by simply sampling the data at very, very dense sort of temporal sampling. And this is obviously a paper that recently came out as part of the package from Hong Kui's group, who have sampled neural development in visual cortex at every stage or every day in development, from prenatal to all the way to the postnatal, all the way to the adulthood. And what I think we've really appreciated, and hopefully you can see this quite clearly in the plot to the right, is that there are many, many different transcriptomic types that are unique to development and they disappear, and then other transcriptomic types that appear during postnatal development. And there are quite a number of transitions. And so this presents one of the challenges that I wanted to discuss today, or at least sort of before we launch into a discussion, which is that, how do we make our interpretation of what's happening there biologically? So you can imagine that let's say that you have a cell type or a cell cluster that is present in high abundance in late postnatal and adulthood, but then has a relatively limited abundance in early embryonic time points. And then you have another cell type, a transcriptomic type that is present very abundant in early development, but then attenuates. Well, our interpretations and our ability to predict and biologically infer what's going on is quite limited, and it currently sort of rests upon two assumptions. So you may have a scenario where these two transcriptomic cell types are, for example, very closely related in transcriptomic space, and there are many cells that sort of transition in between. And this is a case where one would naturally infer that perhaps these are two states that represent a progenitor type, and then which differentiates into post-mitotic cells. But when you have scenarios where the situation is slightly different, and maybe these transcriptomic states are actually transcriptomically very distinct. One possibility is, one inference that you might naturally make, or conclusion you can draw, is that perhaps these two represent actually different cell types, and perhaps one dies or undergoes apoptotic cell death, and another one appears postnatally. But that's not at all necessarily a given truth, because you may also envision a scenario where the transiting states are actually so rare, or the transcriptional changes occur so drastically and quickly that you don't actually sample enough of the transiting cell types to be able to completely rule out the possibility that these two states are not related. And I think, for me, conceptually, there was a lot of intellectual work that came into thinking about these different scenarios, which represent a number of challenges in the field, where transcriptomic states might sort of mislead or obscure our interpretation of the underlying cell lineages. Here is a schematic that represents a few of them from a review by Alon Klein that was published about 5 years ago. But it really nicely, I think, articulates some of the challenges where going off of transcriptomic similarity alone, trying to infer lineage relationships may be quite challenging by itself, and may actually lead to some false conclusion that we have to really reckon with. Thankfully, thanks to the support of Bakken and Brain Initiative, we've been developing, and we and others have been developing technologies such as various types of barcoding methods that allow us to trace and provide some of the ground truth data that we hope to be able to then utilize to refine the transcriptomic predictions, but we can discuss it as well. Now, the second challenge that reverberates, I think, something that Fenna brought about is cross-species comparisons. We knew for a very long time that humans have an expanded diversity of progenitor cell types in the developing, prenatally developing human tissue. And what we're now appreciating, particularly thanks to the work of Aparna Bhaduri, is that many of these transcriptomic cell states that are found specifically in human developing brain that are not found in mice actually then reemerge in certain disease states, such as certain types of brain tumors. And so it's an opportunity for us by looking at development of the human brain to define cell states that might be uniquely recruited for disease progression. And I think that presents both a challenge in how we do that and convince ourselves that that is true, and how to utilize it to develop new therapies, for example. The challenge, of course, is that when it comes to developing human cerebral cortex, there are really limited amount of data sets that are currently available. We cannot repeat the same experiments as was beautifully done by Hong Kui. We cannot sample at every day or even a week of gestation. We really have to limit ourselves to collection of data from a more limited set of samples. However, some of the data sets are already generated, and perhaps we can start working on those. Just a couple of small additional points. Another challenge that I think sampling of developing brain will uniquely position ourselves to do is how to understand the contribution of experience and activity in development. And particularly as developmental processes are protracted in human neurodevelopment, it opens up for certain developmental processes to be uniquely shaped by experience and activity in ways that is not true in other species. And so how does that factor into our understanding and our mapping efforts remains to be seen. We've seen some work coming out, for example, from Larry Ziporski's lab, that mice reared in darkness actually fail to develop certain transcriptomic cell types. I think it's both a challenge in how we design our mapping studies, but also our interpretative challenges, and an opportunity to really fully understand this longstanding question about nature versus nurture in neural development. And I'm running out of time, so I think in the interest of discussion, I just wanted to also point out an opportunity, which is that the developing brain atlas networks taxonomies would be uniquely useful when it comes to community adoption if they could be utilized and integrated into analysis of various experimental models, such as brain organoids, which are beginning to be really utilized across a number of consortia, some of which are NIH supported and by others. And I think thinking about how to make that really a seamless integration, I think would open up a lot of opportunities for community adoption of these atlases. So I'll stop there and open up for discussion. Thank you.
Really beautiful talk, Tom. That was really helpful perspective on some of the opportunities and challenges in the field. I guess my question for you, thinking about what Fenna has already said and some of the discussion that's existed, is, you highlight how we need the resolution of not just the transcriptomic cell types, but also the context of their relationships, and that there's a lot of gaps, especially when thinking about this in humans and non-human primates. So my question for you is, conceptually, what are your thoughts, similar to what I asked Fenna, in terms of how we can fill these gaps and what technologies might be required to be able to do this specifically for the human and non-human primates? And then I'll open it up to the group for discussion.
Yeah, that's a fantastic question. I think, development and establishing relationships and these dynamic frameworks, I think, has been a really unique challenge in single cell biology, because to my knowledge, at least, there's probably been more computational theory development, than the ground truth data to validate that. So I would argue that what we need is more high quality ground truth data in order to be able to make better models, and particularly what Fenna was referring to, predictive models that can help us fill in the gaps. But the challenge right now, I think, is that with different computational strategies, you can predict almost any relationships you might want, and that presents a challenge that, which one do you know is true? And the ones that are not predicted by computational strategies, are you absolutely sure they're not right? And I think that presents really unique challenge and opportunity. And I think if people who generate ground truth data can work more closely with folks who are developing computational algorithms, I think we'll be, we have an opportunity to work together to make that really useful tools for the community that could be applied across a broad range of biological problems.
Thank you. I'll open it up to the panel for comments. I see Honghui has her hand up.
Yeah, hi. Great presentations, all three so far. So I have a general comment, is really trying to integrate everything together and see how you guys think about it. Relating back to the original discussions we had about how to define cell types, right? And how to align cell types across, different conditions. There's the idea of hierarchy and ontology. And there's also idea of how do you even compare cell types? Marker genes, are marker genes reliable or not? I really like the idea of having describing both the ontological relationships and hierarchical relationships. And I think actually selecting marker genes are really critical. And here we're not talking about one or a few individual markers for cell type. We're talking about a combinatorial code of a large number of genes, differentially expressed genes, that you use them together to describe the relationship of cell types. And the key actually here is to select what are the right combinatorial marker genes or code to define. And I feel like, what we're doing now in the adult stage is one snapshot. We can try to do as much as we can, one adult stage from one species, but we will learn so much from evolutionary comparisons and developmental, information. I show some examples that I showed, like, Zhen's, beautiful computational work. The marker genes will be refined. The beautiful specific marker genes in mouse do not work in human at all. But once you align the two, put the two data sets together, you can actually find common marker genes across species. You can also identify more reliable marker genes across development, right? And you find ones that are critical in defining the emergence of a particular type. Even though those marker genes may not be super specific to a cell type, they actually have biological meaning because they are consistent with evolutionary homologies and developmental, consistencies. And I think in the end, if we have all this information together, we'll be much better able to define cell types. Of course, this is going to be a version process, iterative process. But I think, in the end, it will all come together and we learn through the process. Even though the matching of cell types, definition of cell types, clusters are not perfect at beginning, but we will learn so much from development and evolution.
Hungry, I just want to, one thing that you said that I just want to echo, because I think it's so important, is that we think as biologists about canonical sets of marker genes that we use and we sort of trust, but those may not replicate across species or even across technologies. And this is actually very common in immunology, like CD34 is the canonical stem cell marker in human, but it's expressed in the opposite pattern in mouse. And so there's different combinations of cell markers that don't actually overlap with human that together combinatorially define the same functional state. But we have to keep our minds kind of flexible to that idea that it may not always be exactly the same ones that we ascribe to function that may actually be the most indicative and conserved across data sets.
Thank you, Rahul, and thank you, Aparna, for the invitation to give a short presentation here. And what I want to show you is a new spatially resolved single-cell transcriptomics approach that we developed. But I'm also very happy that it actually integrates pretty well with this last topic about integrating spatial transcriptomics or spatial omics data. And so actually before going into the specifics of this approach, maybe because of the nature of the session, let me just actually mention very briefly the different spatial omics approaches. They fall largely into two classes. One class is based on multiplexed imaging, and that has very high, really excellent spatial resolution, not only single cell resolution, but also subcellular resolution. But this class of approach typically requires a pre-selection of a gene panel or targeted gene panel. And then another class is based on next generation sequencing. And that doesn't require, it's an untargeted approach. It doesn't require pre-selection of gene panel, but doesn't have as high of a spatial resolution as the multiplexed imaging approach class. So, so then, there's a major gap to fill, and one could try to fill it by, for example, making the imaging approach to hit the whole transcriptome wide scale, or making the next-gen sequencing-based approach to have a higher spatial resolution. And I will talk about is making the imaging reaching the whole transcriptome scale with isoform resolution. And this approach is based on the MERFISH technology that we developed. And I'm not going to go into any detailed introduction of MERFISH because many of you have already used it and then know what it is. I'll just say that it is an approach based on, using robust barcoding, combinatorial labeling, and sequential imaging to allow genome scale imaging. And we and others have used that to construct a spatially resolved cell atlas of the brain. For example, I won't mention our work, but you've also heard of really fabulous work from Allen Institute. So then if in thinking about how to make the MERFISH approach to enable whole transcriptome imaging, the MERFISH itself really has the multiplexing capability, meaning that it's not the number of genes or transcripts that we can image that is limited, because we can reach, because the exponential scaling, we can image a very, very large number of transcripts relatively quickly. But a major challenge is that some transcripts are very short. And so because of that, it doesn't generate a sufficient signal for high quality multiplexed detection. And also some transcripts might not be short, but for example, different isoforms of the same gene can differ by a very short sequence. So that again requires the ability to actually measure either short transcripts or very short sequence difference between transcripts. So that means we want a powerful amplification method. So we developed a in situ RNA amplification method that is based on reverse transcription and followed by transcription. So take a transcript, for example, a messenger RNA, but it could be other kinds of RNA too. Then we have a primer, in this case, a primer that binds to the poly A tail of messenger RNA that can serve as reverse transcription primer that can actually do reverse transcription and generate the complementary DNA. And then at the end of the reverse transcriptions process, a few nucleotide will be added, defined nucleotide will be added. And that allow us to bind a template switching oligo to it. And then this template switching oligo also includes a T7 promoter sequence. So with that, the reverse transcription will go on to completion and then also copy the T7 promoter. And that allow us then to do in vitro transcription to convert a single copy of complementary DNA into many, many, many copies of RNA and hence achieve a very large amplification. And then these then generate in situ amplicons. Each amplicon represents an original single RNA molecule. And then we detect that with MERFISH. So I went through the method a little slowly, but I realized this is supposed to be a very short talk. So I'm going to go through the rest very quickly just to show you capabilities. For example, we can indeed do whole transcriptome imaging. Here we're imaging more than 20,000, 23,000 genes and yet still achieve a very high quality measured by a very good correlation with bulk RNA sequencing and excellent replicate reproducibility. And then with that, then of course, now we can do spatially resolved single cell gene expression profiling at the whole transcriptome scale. Then we can then build a molecular atlas of, for example, the brain, and then looking at the gene-gene correlations and then find gene modules that have a similar spatial expression pattern. And then with that, actually, identify a molecularly defined brain regions, which is not only what we find to be very helpful for actually registration into common CCF, but also through, small functional regions that have co-expressed genes and some of the very interesting functional genes to actually understand the molecular mechanisms of the function of these brain regions. I do apologize for the lack of details because we're running short of time, so I'm very conscientious on time. Of course, we can also do cell typing. And then because now we have each cell to have whole transcriptome scale expression, then we can also do a very comprehensive spatially dependent DE gene analysis to see the same cell type, how are the gene expression different from region to region. And one of the questions that we do have in this particular section is whether we should use spatial location as information for different cell typing, or whether the same cell type can have different gene expression. And this approach has certainly helped illuminate on that front. And then also because we have a single cell expression profile that's spatially resolved at the whole transcriptome scale, it also allows us to do much more comprehensive cell-cell communication analysis, ligand receptor analysis that are based on both spatial proximity between cells and gene correlated gene expression, for example, co-upregulated ligand receptors. I'm not going to go into any details, so with the time in mind. I will just slow down just slightly to talk about this last aspect, which I want to talk about, which I'm very excited about, is that the fact that we can measure things at the whole transcriptome scale and distinguish very small, very short sequence differences that does enable us to do spatially resolved single cell isoform profiling. And we know that individual genes can actually express into different isoforms that have different functions, and then some diseases are actually dysfunction at the isoform level. So this is very exciting to us. So as a proof of principle demonstration, you can see that in addition to the 20,000 genes, we also imaged 10,000 isoforms. So in this this first proof of principle work, we tried to demonstrate a very large number already. And I won't show you all, of course, all the very interesting things that we learned about brain region and cell type specific isoform usage. But just to tell you that with this technology development paper, we actually really remarkably identified a very highly spatially diverse and cell type specific isoform usage. I do want to use one example. I'll just illustrate one example. This is a very interesting gene, MEF2C, which is its protein coding form actually codes a calcium-dependent transcription factor that are important for neurodevelopment. And then, if I zoom into these two different isoforms, so this 218 isoform is actually a protein coding isoform. And then we see a very interesting spatial and cell type specific distribution in the cortex and dentate gyrus. And the location is actually, and cell type specificity is very consistent with its known function. But also what's this other isoform is very interesting. It's actually a non-coding isoform, and its function is unknown. And when we looked at its spatial and cell type specificity, it's highly complementary to the protein coding isoform. For example, the protein coding isoform is not expressed in the thalamus hypothalamus region, but this non-coding isoform is expressed. And then if you look at the cortex, you might think that, hey, they both are expressed there. But if you look at the cell type, it's very interesting. The coding isoform is only expressed or highly enriched in the excitatory neurons, while the non-coding isoform is highly enriched in the inhibitory neurons. So it forms a very complementary expression pattern, which actually suggests a potential regulation mechanism. And that is this non-coding isoform expression pattern actually regulates the expression, the coding isoform's expression patterns through competition. And this was not known for this gene before, but for some other genes, it has been suggested that non-coding isoform has this regulation capability of regulating gene expression by competing, for example, spliceosome splicing machineries and so on. So we also found brain regions that are particularly enriched for very specific isoform usage, which I won't go into details. But I just want to conclude here by saying that, as I said, this approach now we're very excited about it because it not only now allow us to do more comprehensive spatially resolved gene expression profiling, but also enable us to do spatially resolved single cell isoform profiling, can help us better understand the molecular mechanisms of not only normal functions of the brain, but also dysfunctions in diseases, but of course other tissues. I'll just highlight that the lab members who are leading this work, Limor Cohen, Eran Halpern, and Tim Blosser, and open up to questions not only about this talk, but also about this general topic of integrating spatial data.
All right, thank you, Xiaoyin. That was beautiful. It's striking how the underlying molecular technology are starting to converge between the sequencing-based workflows and the imaging-based workflows. So sort of RT-T7 has been something that people have been doing single-cell sequencing readouts for a while.
That's right, dissociated single-cell sequencing, exactly, right.
And you're doing it all in situ and then reading out the amplified RNA, which is wild. I also want to, Aparna had to leave to teach, so I want to thank her. I'll thank her more at the end, but I'll run the Q&A. I wanted to ask one question just to get things started. I mean, over the last maybe sort of decade or longer than that, you sort of progressively knocked off one challenge after another in spatial analysis, whether it's, increasing the number of cells you can image, or obviously increasing the complexity of the readout, and now getting down to even isoform levels of resolution. You and others have had papers on other modalities as well, and including, chromatin-based modalities and RNA binding modalities. As a provocative question, sort of what's left? What do you I mean, there's so much you can do now with spatial analysis that we sort of couldn't dream of a decade ago. And obviously there's a lot of work to sort of make these commercially available, but in terms of next sort of research frontiers, I'm curious what you're excited about, if that's an interesting question to start with.
Yeah, I think it's a really, really excellent question, and that's what we all think about. And I do think that there are two things that I want to mention. Both are function related. One is obviously for all of us who are in this audience, think about this cell atlas, this extraordinary diversity of cells that we identify and map their spatial locations. Really, a very important, obvious question is what's their function? How are they functionally different, and so on? And because of that, I think multimodality is very important, especially if we can, consider the modality that connects the molecular and spatial signatures to the function, to combine with functional imaging such as calcium imaging, and in the nervous system case also connectivity of neurons because the circuit is so important for function. And the other also about function is not only at the cellular level, but we're all very interested in getting down to the molecular level, both for fundamental understanding, but also for disease. Things goes wrong, and when you want to do therapy, and it usually happens at the molecule level. So what's the molecular mechanism of functions? And there, perturbations such as gene editing, CRISPR, and so on are very very important. And then we and others have been working on doing functional genomics, meaning that we can perturb at a very large throughput genome scale and measure, hopefully, also at this very high throughput spatial, genome scale, functional scale, and so on, so that in one experiment, we hopefully can generate a huge number of hypotheses of the molecular mechanisms of functions of genes by not only perturb at the genome scale, but do all kinds of imaging-based, sequencing-based measurements of all kinds of functional properties and morphological properties, molecular properties, and so on.
Your work with Jonathan Weissman certainly comes to mind there. That's very exciting.
That's what I have in mind when I've been talking about this. Although I didn't mention that we do have this recent perturb multi work that we do this both by using Merfish and perturb-seq to do, but other labs do that too. So it's a really interesting new area. Also links to the a lot of effort of virtual cells, as many of us know, right? And then also it links probably very well to the next topic, because for virtual cell, then computation is exceptionally important as the work that you do and others. And then there's a machine learning section that come up, so all these things are linked together. We're living in a very exciting period of time.
Margot, do you want to jump in?
Yeah, my question, Xiaoyin, was how quantitative is this method? I ask it because of the reverse transcription and amplification step.
Yeah, that's an excellent question. I'm sorry, in rushing through time, I didn't mention that. So I mean, thank you so much for mentioning it. So it is quantitative because one of the things that we characterized about this is when people try to do, especially when you think about isoforms and so on, you really do not want to have end bias, meaning that you want to amplify the whole transcriptome uniformly across the entire length so that you can be quantitative. And we did characterization and then found that our approach do not have end bias. So that enables quantitative analysis.
Thank you.
I think we have 5 minutes left, so let me pose one more question that maybe others who build common coordinate frameworks also want to jump in, and then we'll go to summaries. But I think when you talk about integrating data, which this session is about, when we're talking about spatial data, you also have this incredibly interesting problem of anatomical registration and spatial registration, in addition to molecular harmonization. And so much of the mouse data has been, as far as I can tell, sort of very seamlessly integrated together using the common coordinate framework from the Allen. I'm curious, as you do more and more human work, how challenging that is for you, and what efforts there are sort of to create these reference anatomical frameworks for other species as well. Anyone can jump in.
So I can answer while others are thinking about answers too. Less of an answer, but comments, and then hopefully leading to more questions. I think when we were doing CCF registration, we do find that the molecular signatures that we can image and then generate molecular maps and then generate molecularly defined regions that really help us a lot in refining the alignment into the Allen in CCF. That said, that benefit a lot from when we're imaging a whole mouse brain, a lot of features, if you have a lot of variations and so on. So I also have the same question as the one applied to human, especially if one image a sub-region of a human brain. Is the feature significant enough for us to do an accurate registration or not? I'm equally curious about, people who have experience on this front.
I'll wait for anybody to jump in on that. I think that's probably one of the most interesting questions and sort of cutting edge frontiers for spatial integration. Go ahead, Ankwi.
Yeah, I'm happy to jump in. I think the CCF, especially for human and non-human primates, will be really the next challenge and frontier, because it's been extremely useful in the mouse. But how to define CCF in the primate brains? And I think we will need to have, really high resolution and high sensitivity spatial data. I'm very excited about the new technologies Xiaoyin talked about, because, we try to do imputation, right? Imputation of RNA-seq data into the space, trying to capture all the potentially subtle gradients, and not only discrete, the cell type, cellular distinctions, but maybe even gradient distribution of things. Imputation is always, Rahul, very well, challenges, right? So if you can have direct, high sensitivity spatial data, it will help to define brain anatomy so much better, especially thinking about the different cortical areas, right? We still don't know if there are distinct transcriptomic signatures of different functional cortical areas. So you'll be very, and of course, subcortical, we know so little because we don't have that much of the anatomical and connectional data for the primate brains. So I think that's a great question, and it would be, something very interesting to work on.
I think the point that those maps will benefit from molecular data in the way that the mouse one couldn't because the molecular technologies weren't available is very, very powerful and might overcome the much greater degree of phenotypic variability that there is across human and non-human primate samples compared to the mouse. But the cell types exhibit a much greater degree of conservation, and so it can be used to facilitate alignment there. I like that very much.
Yeah.
Okay, I also wonder a little bit that, because when imaging, ultimately in the future, I don't know when, it would be great to image the whole transcriptome, like what I show here at the whole mouse, human brain level and so on. And of course, the amount of imaging burden that's required, eventually, maybe we overcome that. But in the meantime, maybe that also links to the next section, is whether there is an approach that we can use this and the more simpler imaging approaches, such as, DAPI and other kind of, signatures and so on, so that one can do machine learning based training so that, you can get this sort of level of information, at least to help with the CCF registration across labs, right? So that not every lab has to do this very highly multiplexed imaging for the CCF registration purpose.
Okay, I think I said we would wrap up by 2:45, so I just really want to thank our speakers, everybody who jumped into the discussion, Aparna for co-leading this with me, and the NIH for inviting us. In the interest of saying sort of one concluding remark, I think what was really remarkable about this session is how much everybody really is already using these reference frameworks, reference data sets, and atlases to make sense of their data, not only within RNA, but also across modalities, across developmental time points, and across species. The biggest theme that came out for me across these talks was, as a next frontier, the ability to use very complementary data, functional data, lineage data, ground truth data, as Tom argued for very persuasively, to be able to understand, how finely resolved should we annotate these cell types? When can we really trust matches across species and across developmental time points? The matching of marker genes is only a very sort of scratching the surface way of being able to argue for robust science there, and complementary data, which is harder to generate at scale right now, but isn't impossible, will play a really key role in sort of understanding the strengths and limitations of different atlases. So I like that very much. Okay, so with that, thanks everyone for working through the technical difficulties, and I'll sign off.